Proteins are the essential biomolecules that carry out most chemical and mechanical processes within living organisms. Since the structure of a protein determines its function, protein folding, which is how proteins form their three-dimensional shape, is one of the most important subjects in chemical and physical biology. Proteins mostly fold spontaneously in cells. Therefore, we can model the ability of proteins to fold into certain structures using the total difference in Gibbs free energy between its unfolded and folded states. The more negative the delta G, the more stable the folded structure. This, however, raises an important question. Since Gibbs free energy is partly dependent on entropy, and a protein that is folded has less possible microstates than an unfolded protein, how can protein folding ever be spontaneous? It turns out that the Gibbs free energy contributions to protein folding are numerous and complex. Besides the unfavorable contribution from conformational entropy, there are also favorable contributions from mainly the hydrophobic effect, which is also entropy driven. In pure water, water molecules are constantly forming and breaking hydrogen bonds with one another. However, when a hydrophobic molecule is added to the solution, the water molecules around it are forced to create a semi-rigid cage of water molecules around it. While this is a net decrease in entropy, the presence of multiple hydrophobic molecules can actually increase the overall entropy. Hydrophobic molecules in aqueous solution will tend to pack themselves tightly together, forming a core that has no water inside. Forming this core decreases the total hydrophobic surface area, decreasing the amount of water molecules required to form the cage around it. This consequently frees some water molecules to roam around the solution, which is an increase in the number of microstates of the system, and therefore overall entropy increases. Because proteins consist of a sequence of both hydrophilic and hydrophobic amino acids, the hydrophobic effect is a strong driving force behind protein folding.